Okay, I think we're going to get started. I wanted to thank everybody for joining the NINDS Biomarker Program Q&A webinar. The way that this webinar will run is we will do a short presentation on the program and go over the funding announcements. And after that, we will have a Q&A with everyone. Just first announcement, this meeting is being recorded. And the hope is that we will be able to send this recording out to everybody by the end of this week, along with the slides. So everybody will get a copy of the slides, and assuming this recording does what we hope it does, we will send that out to you. The way that you're going to ask a question, everything will be done via the Q&A box. Please use the chat box for any kind of technical questions you might have to me, but ask all questions in the Q&A box. If you don't see the Q&A box in the lower right-hand corner of your screen, click on in the lower right-hand corner of the screen, click on the three dots, and you will see the option to open up the Q&A. Once again, use the Q&A for all questions. The chat function is just for tech support. So with that being said, I'm going to pass it over to Program Director Marianne Pellebaunter.
Hi, thanks, Tim. Good afternoon, everybody. And again, thank you so much for joining us in this webinar to hear about the NINDS Biomarker Program. The program directors are myself and Carol Taylor-Birds, who will also be presenting part of this webinar. And then our health program specialist is Genevieve Lind, and she will be helping with questions and answers as well. So the first thing I want to impress all of you with is that you might say biomarkers are talked about a lot, but what do they actually do? This slide gives you an idea that biomarkers actually can improve the probability of successful drug development. If you look at the graph on the left, that shows that if you have a pharmacodynamic biomarker that shows proof of mechanism, it really does reduce the probability that the drug candidate will fail. So you can see that if you have proof of mechanism, roughly 31% of drug candidate programs would fail. But if you do not have a proof of mechanism, 74% actually fail. So that's a big difference, and really does argue that having a way to at least know whether the drug met its target makes a big difference in whether or not the project will go on. Similarly, patient stratification biomarkers that allow you to make smarter, better designed clinical trials so that you're actually assigning people to groups that make sense. They also improve the probability of success. So in this particular graph that was done by the bioindustry analysis with D.W. Thomas as the first author, you can see, especially if you go to the phase 1 to approval, which sort of summarizes the whole graph, with the blue bars, you can see 25.9% of projects actually had a very good probability of success, whereas only 8.4% did if they did not have that patient selection biomarker. So again, with patient selection biomarkers, 8.4% probability of success. Without biomarkers, 8.4%, and with 25.9%. Go to approval. And another thing that I think is important to figure out right away is just to be aligned on what we mean when we're talking about biomarkers. Even the term biomarker is very loosely defined, and a lot of people have different definitions. In this program, we try to be extremely consistent with the FDA NIH definitions from a glossary called the BEST glossary. BEST stands for Biomarkers, Endpoints, and Other Tools Resource. Biomarker is, according to that glossary, an indicator of a normal or pathological process or of a response to a therapeutic. Context of use is something else we use a lot, and we'll talk about a little bit more, but it's the manner and purpose of the use of a biomarker. If we talk about things that are fit for purpose, And a lot of this fit for purpose has to do with how much validation you need to actually use the biomarker. So as I said, it refers to the required degree of validation for the intended use of the biomarker. You're going to hear about analytical validation, and clinical utility later on, but just quickly, analytical validation really establishes the performance characteristics of a measurement that in terms of how acceptable are those performance characteristics in terms of sensitivity, specificity, accuracy, precision, and other relevant characteristics. Clinical validation establishes that the biomarker acceptably identifies, measures, or predicts the concept of interest. And finally, clinical utility refers to the idea that you can conclude that the given use of the biomarker will lead to a net improvement in health outcomes or provide useful information about diagnosis, treatment, management, or prevention of a disease. This slide was put in here just to give you an idea of how this program views the process of developing a biomarker as you move from discovery through FDA qualification. The blue wedge here gives you an idea of how much validation evidence you need as you move through those levels. So in discovery, usually the types of activities that occur are identifying the biomarker itself, standardizing the sample source, obtaining pilot clinical validation of the new biomarker other than just correlations with the clinical concept of interest, and then developing the detection method. In analytical validation, the context of use must be established, whereas the context of use, I should mention, in discovery, there should be a hypothesis, but it doesn't necessarily have to be established yet at that stage. In analytical validation, pre-analytic variables are identified and dealt with. And as we mentioned before, this is where you establish the acceptability of the performance for your detection method. In clinical validation, these are, you're assuming now you've analytically validated the biomarker and you have it, but now you need to do large prospective or retrospective clinical studies. In general, during this stage, you evaluate the overall utility for clinical practice and for drug development and clinical trials. Things that you're looking at in clinical validation studies include clinical sensitivity, clinical specificity, positive predictive value, negative predictive value, and you're looking at classifiers or predictors. You can be looking at binary variables like a diagnosis, yes or no, you have it, or how severe is the trajectory of the disease going to look over time. Qualification is something that it's a program that the FDA has, and this actually is it's a very nice program, committee based within the FDA, not necessary for these grants, but again, we try to be as consistent as possible with the requirements of this program so that if you decide you want to take your biomarker for FDA qualification, you won't have to do a lot of things over. So basically, qualification says that within the context of use, the biomarker you have allows specific and reliable interpretation. And once you have a qualified biomarker, you would also have a plan to integrate it into clinical trial design or clinical practice. A little bit more on the context of use. It's a concise description of the biomarker's specified use in therapeutic development, and it relates to the type of biomarker. And this can be found also in the BEST biomarker resource. It tells you about the biomarker category and the biomarker's intended use in therapeutic development. So some examples are you might want to have a biomarker that stratifies or enriches patient populations. Another context of use is to demonstrate therapeutic target engagement. Another to indicate the risks for developing a disease or condition. And a final one perhaps, and these are all just examples, to establish the response to a therapeutic that would assist with dosing decisions. And so a critical point here is that your experimental design will be evaluated relative to your context of use. So we can't emphasize enough how important in your application it is to have a little section that actually details what your context of use is. Even in the discovery phase, at least it should outline what your hypothesis for the context of use is. And as we mentioned, it's important that we do things using the FDA's guidelines that are out there. The biomarker program, again, is designed to be consistent with them. So there's an evidentiary framework for biomarker qualification that is available actually using the link that's provided in this slide. And just to give you an idea of the types of biomarkers that are outlined in the BEST glossary, these are the basic ones: pharmacodynamic response, diagnostic, prognostic, predictive, susceptibility, risk, monitoring, and safety. And each of these are very carefully defined in this glossary, but just to quickly go over, pharmacodynamic response biomarkers reflect exactly what the biomarker, what the drug is actually doing to the body. So you could, for instance, if you have a biomarker that is something like receptor occupancy, that would directly show that the drug has occupied the receptor as it was planned to do. And you could show that in a quantitative concentration dependent way. If you were looking at a diagnostic biomarker, that will give you an idea of whether or not a disease is present, or it might give you an idea of how to diagnose subtypes of a disease and separate them, which would help in a clinical trial and would also help in treatment decisions in clinical practice. A prognostic biomarker will predict the trajectory of a disease, and it might actually also sort of be paired with a diagnostic use, in that it might not only diagnose whether the disease is present or not, but it might also predict whether disease severity is going to change or the phenotype of the disease is going to be one way or another. Predictive biomarkers predict whether or not the patient will respond to a particular therapeutic. So a therapeutic's involved there. Susceptibility risk predicts the risk in normal people to whether or not they would become diseased. A monitoring biomarker allows you to monitor the effects of a therapeutic, like for instance in cancer. Safety biomarker allows you to predict or monitor potential toxicity of a therapeutic. So our program at NINDS has a goal of facilitating the development of high-quality biomarkers for neurological and neuromuscular disorders to improve the quality and efficiency of clinical research. And this is our act, this goes along with the mission for NINDS, and we should emphasize that applications that come in for any of the FOAs in our program must meet the mission of NINDS. The resources we have are funding opportunities, which we'll go into in a minute. And then they also must support a rigorous fit-for-purpose validation process. So again, your application, that's where, saying what your context of use is a very important aspect. And then the method and the approach you're proposing should make sense relative to what you're going to use it for, and should provide that degree of validation for your intended use. We also have quite a set of tissue and data repositories as resources. We have repositories, different ones actually, that provide samples, including blood, plasma, saliva, CSF, tissue cells, DNA, RNA, and also our repositories for data. And you can find out more about the separate repositories by clicking on this link, and this leads you to our general biomarker program site. So this lists the funding opportunities that we have that support discovery and validation of neuroscience biomarkers. You can see the first one, PAR 19-315, supports the discovery stage of biomarkers that meet the NINDS mission, and it's a phased grant, an R61, R33 mechanism. Now, the next four that you have, you can see that they have 0XX as their number, and what that means is that the validation FOAs have now expired, and so we're in the process of reissuing all of these. So we can't exactly give you the number or the exact submission date because we don't have that information yet, but it should be quite soon. So I would hope that all of you, if you're interested in this, just keep in mind that we definitely intend to reissue, and that process should be finished very quickly, and they should be published soon. So there are two for analytical validation of a candidate biomarker. One is an academic U01 mechanism, and the other, the U44, is an SBIR or STTR mechanism. Because these are used, all of these are used, actually, they're cooperative agreements, so they're different from the phased mechanism. The last two are clinical validation, either the academic model or the SBIR version of the grant mechanism. And that, just to point out, the next submission date for the discovery PAR, which is not expired yet, is February 22nd, 2021. So just to go into the mechanisms, the R61, R33, as I said, was phased. The R61 phase is typically 1 to 3 years. Most people do 3 years. The budget's not limited, but it must reflect the actual needs of the project. The R33 mechanism, which is the second phase, can be 1 to 2 years. And again, same thing with the budget. Now, speaking of budget, if your application requests $500,000 or more in direct costs in any particular year, then you must contact us at least 6 weeks before submitting the application, because we have to get NINDS permission to review that. So that's how, we are sort of, that's how we look at things that are getting a little bit more expensive than $500,000 in any single year. The other thing to point out is that the total project duration is no more than 5 years. The other thing I want to mention too, finally, is that even though there are 2 phases to this R61, R33, the application must include both phases. So your application should outline what you're going to do in R61 and outline what you're going to do in R33. The other mechanism, the U01 and U44 cooperative agreements. The U01 length is up to 5 years. The same thing with the budget's not limited, but must reflect actual needs. The U44, which is the SBIR cooperative agreement, first of all, should point out we only use the fast track mechanism. So we don't use the, the other phased mechanisms, just fast track. Phase 1 of this fast track is up to 2 years. Phase 2 of the fast track mechanism is up to 3 years. Money allowed for, budget allowed for phase 1 is up to $700,000 per year. For phase 2, up to $1.5 million per year is allowed, and it must be reasonable and appropriate. For the U01s, same kind of thing. If you are asking for $500,000 or more in direct costs in any year, you must contact us 6 weeks prior, because we do have to obtain permission from NINDS to get your application reviewed. That rule does not apply to the U44. So the other thing I wanted to point out about these mechanisms is that both of them, whether they're the phased grant or the cooperative agreements, are separated by milestones. So to go from the R61 to R33 phase, there has to be one set of go, no go milestones. These milestones should be in your application, clearly outlined that these are the set of milestones you intend to use to go between the 2 phases. They should be clear, they should be quantitative, and they should be definitive. So milestones that just reflect a set of activities are not appropriate. So these milestones have to be things that you need to see in order to make an argument that it makes sense and it is feasible to go to the R33 stage. And transition occurs via an administrative review, so it doesn't have to go back into the regular review cycle. For the U44, here, one of the big differences is that you have to propose milestones for each year of the grant. So between year 1, year 2, and so on. Same kind of idea, though. They have to be go, no go milestones. So again, a decision has to be made based on these milestones, which is why it can't be just a list of activities. These milestones have to be very clear. They have to be quantitative. And again, you have to, one way to look at it is your milestone showing a showstopper. Like if you didn't recruit X number of patients, you wouldn't be able to go on. It's that type of milestone. Or you might be something like you need to have a certain range in your area under the curve with a receiver operator characteristic analysis for sensitivity and specificity. So very quantitative, where we can objectively make a decision. And again, transition occurs via an administrative review. So now I want to just say a little bit about the discovery FOA. And with this one, the goal here is to deliver strong candidate biomarkers or signatures for neurological and neuromuscular disorders that are ready for definitive analytical and clinical validation studies. The entry criteria are, can't emphasize enough, what you're proposing must be consistent with the mission of NINDS. So especially in terms of the disease area. You have to have a cogent biological rationale that includes available supporting preliminary data that supports the feasibility and makes a good argument for why this is going to be important, this biomarker, that it's not just an add-on, that it's really going to make a difference. You have to make that argument. And another thing is it needs to have relevance for therapeutic development or making decisions in clinical practice. And then another thing that's important is your team. There should be a demonstration in the proficiency for the biomarker modality proposed. And here I want to point out that we are pretty agnostic to biomarker modality. So we don't care if it's imaging, physiological, genomic, proteomic, whatever. We just want to make sure that you're developing the biomarker in such a way that you will actually have a deliverable when this is over, and the biomarker could be used in the community. Okay, application characteristics are things like, so this would be what you would see in an application for the discovery FOA. So strong justification for the use of the proposed biomarker detection method, as I mentioned before. Your application should address the feasibility of the biomarker, including evidence that you've thought through potential added clinical burden. Is it feasible to actually do in patients with the disease? And cost, is it something that can be afforded? There should be a context of use hypothesis proposed. We tend to prefer that you focus instead of having many contexts of use. It's usually better, makes a crisper application and approach if you focus on a particular type of context of use. And then the application should contain proposed studies to identify the biomarker and verify that it reflects the clinical endpoints that are consistent with the context of use. Applications usually contain studies that are focused on the development and initial validation of the detection method as well. So we look for not only identifying the biomarker and providing that initial biological/clinical verification, but also that you've done some work to develop the detection method. And then the studies should be present that provide preliminary validation of that biomarker. For instance, let's say you identified a biomarker and developed a detection method in the R61 phase. Then in the R33 phase, it would be appropriate to do a pilot level preliminary validation of the biomarker with its new detection method that's consistent with the context of use. And with this preliminary validation, you would have variables related to the clinical sensitivity and specificity of the biomarker. Other things that's really important is you have to have these milestones. We can't emphasize enough. We can ask you to withdraw your application if there are no milestones. And they should describe the metrics for evaluating initial biomarker and detection method development. And then it's also important that the biomarker signature categories and descriptions reflect that FDA NIH BEST glossary of endpoints. So this is something that if you're interested in applying, just check this out, use this link and get that best resource as soon as you can if you want to apply. That way you could always refer to it and make sure everything's in sync. And then the research strategy clearly describes how the application will utilize a rigorous design, execution, and interpretation of the proposed studies. Responsiveness criteria are, basically, again, emphasizing that you're identifying and conducting some preliminary validation biomarkers that indicate This is an example, pharmacodynamic responses to a therapeutic. And I mentioned before target validation and things like receptor occupancy, but more indirect pharmacodynamic responses are fine too. You just have to show that you have really solid validation that they still reflect how the drug is affecting the target. You can also be looking at prediction of efficacy or safety response and looking at diagnosis, prognostication, and risk susceptibility for the disease or disorder. So these are examples of responsive studies. Things that are not responsive are biomarkers that are not in the mission, within the mission of NINDS, or if they're natural history studies that are aimed at only understanding disease pathophysiology in the absence of biomarker identification. So again, here you may have to do some natural history work, but as long as the major intent of the proposal is biomarker development, that's what's important. It is also not responsive to use non-standardized sample sources. That has to be, in your approach right up front. And then it's also important that there's therapeutic target identification is set up to identify a neurotherapy. So actually, I said that wrong. So it's not responsive to try to come up with a new therapeutic target in the absence of trying to develop a biomarker. And it's also not responsive to have preclinical animal studies without a transition to studies that involve human samples. So that's another thing. We do need to see, at least by R33, that you're using human samples. It's okay to use animal data in the beginning to first identify the biomarker, but you have to translate to human samples during this particular, even during the discovery FOA. It's not responsive to develop candidate therapeutics as the primary intent, and it's not responsive to look at efficacy testing as the primary intent. And of course, you have to have milestones. Okay, so that's my part, and I'm going to hand it over to Carol Taylor-Birds, who will tell you about the validation FOAs.
As soon as I unmute myself. Thank you, Marianne. So as Marianne said, we're hoping these will be out on the street very soon. And this is the reissue of the analytical validation PARs that I'll go over, and then the clinical ones next. So the goals of these UO1 and U44 PARs are to enable the investigators to conduct the rigorous analytical validation of the biomarker, evaluate the detection method, and then establish the performance characteristics of the detection method. And the entry criteria for applying to the analytical validation PARs are that it's within the NINDS mission, of course, that the context of use has already been identified, that the biomarker has undergone the initial clinical validation, just as Marianne was talking, would be established in the discovery phase, and that the method of detection has already undergone initial development. And what we mean by that is, it wouldn't be appropriate for something to come in to develop a brand new device or assay for the analytical validation. It needs to already be developed. And so some of the critical aspects of the application should include the strong justification of the biomarker and the method of detecting it, and of course, what the unmet clinical or research need is. You should also really directly address feasibility, including any potential added clinical burden or cost that's associated, that might be associated with using the biomarker or important for the detection method. So you want to be sure to describe what the status of the existing detection method is and how it will be optimized for the context of use that's being proposed. And that would, include things like laboratory or point of care settings. And also note that the expectation for the analytical validation should be conducted in a multi-site setting. So if you're going to only do a single site, you can do that, but you really need a strong scientific rationale for why that would be appropriate. But the expectation is multi-site. Also, as Marianne discussed earlier, you'll want to be sure to describe the biomarker using the definitions used in the FDA NIH BEST glossary. And your research strategy needs to clearly describe how you'll use a rigorous design and should spell out the analysis and interpretation to be conducted really thoroughly and clearly. And finally, just a reminder that these are milestone-driven applications, as Marianne talked about before. So you'll need annual milestones, because unlike the R61, R33, this is milestones set each year. And these need to quantitatively describe the critical steps and results that are needed at the end of each year to move the project forward. And those should include things like the performance characteristics that need to be met. So the metrics for the analytical validation should include accuracy and precision of the detection method, along with the sensitivity and specificity and the dynamic range. And then depending on the biomarker, you might have additional metrics like the dilution linearity and standard linearity and parallelism with clinical samples. And then establishing the reference intervals, controls, and calibrators can also be important analytical validation metrics. And next. So responsive applications for analytical validation PARs include applications that plan to validate the biomarker detection method, and that includes, pharmacodynamic, prediction, safety, diagnosis, basically agnostic to the category of biomarker. But non-responsive studies would be those that, just like the discovery, that the sole purpose is to identify or to develop the therapeutic agent as the primary intent, or evaluation of clinical efficacy, clinical management as the primary intent. And then again, for these PARs, both these and the clinical, animal models are not appropriate and in vitro models are not appropriate. These should all be using clinical samples, data, or clinical studies. And then studies that fail to include milestones. And if any of these are in the application, they can be administratively withdrawn. So moving on to the clinical validation. And here we're providing the opportunity to conduct a rigorous clinical validation of the biomarker for its context of use. And researchers are encouraged but are not required to work with the FDA for biomarker qualification. Marianne covered that a bit in the beginning. So like any of these PARs, these applications must be within the NINDS mission. Here, the context of use is especially critical and should be really clearly defined. And the biomarker must already be identified, but now the detection method must also have been analytically validated already, and there should be evidence of that in the proposals. And there should be a plan for managing the pre-analytic variables, and the initial clinical validation, of course, should have already been demonstrated as well. So the characteristics needed for clinical validation application are the same as those in other FOAs, including a strong justification, addressing feasibility of incorporating the biomarker into research or clinical use. But the study designs need to definitively test the clinical sensitivity and specificity to the clinical populations that the biomarker is intended to be used with, right? So the outcomes to validate the biomarker must be clearly stated and justified. And like the analytical validation PARs, these should be multi-site study designs, again, unless there's a really strong scientific justification why only one site is possible, which can come up in certain rare disease conditions, for example. Of course, the biomarker category should be defined. A rigorous study design is critical, and the statistical plan and interpretation of the results need to be really clearly laid out. And again, annual quantitative milestones must be included. So clinical validation metrics should include sensitivity and specificity of the biomarker within the proposed context of use. And metrics should include receiver operating curve to establish the appropriate cutoff or threshold to making decisions with the biomarker. And the prevalence of the biomarker within the patient population that is described in the context of use needs to be estimated so that you can provide the positive predictive value and negative predictive value by the end of the project. So responsive applications include those whose goal is to clinically validate any biomarkers as already described. And then again, non-responsive studies include those where the primary intent is on testing the therapeutic device, clinical efficacy, et cetera. And again, of course, animal models, in vitro models are failing to include milestones are grounds for administrative withdrawal. So just to sort of summarize, one of the things that investigators, we have a lot of investigators who have questions about whether they're ready to come into the program. And so what you really want to do is be able to go through these questions and answer them and see if you're at that readiness level. And so do you have a clearly defined context of use for your biomarker? Because this is probably one of the things that's most common, is not having a clear context of use for what you want to do with your candidate biomarker. And then what category of biomarker is it? Use that BEST glossary to really look at those definitions and make sure that you're describing it in the appropriate way and that your approach matches, can actually demonstrate its reliability for that context of use and for that category of biomarker. And then how will it be used in research or clinical decision making? What is its value? What populations can it be used with? And then what's the unmet clinical and/or research need for your biomarker? Why is this so important? And how it will be useful? And if validated, how will the use of the biomarker compared to current practices or available tools? So this is another, subset of questions here you want to think about. Is this going to be more sensitive and specific to the context of use that you're proposing? Is it something that would be easier to use or implement? Will it be more cost effective? It doesn't need to be all of those things, but you want to think about how it fits within the framework of what's already available. And then, of course, we can't emphasize rigor enough. Your approach absolutely has to rigorously and thoroughly test your biomarker for the context of use. And we've said that many, many times over the last few minutes. So a few more questions just with the specific PARs to consider. So for the discovery, by the end of the 5-year proposal, will you have a candidate biomarker endpoint with a developed detection method and a preliminary validation in human samples by the end of the grant period, right? And then for the analytical validation, have you considered and addressed all major sources of sample variability, the pre-analytical variables, and overall performance of the detection method? Well, that has been established. How reliable is it, and how reproducible is it? And can the detection method be reliably used by others? Again, part of the reason for that multi-site, how critical it is for the multi-site, is you really want to demonstrate reliability and reproducibility. And then finally, for clinical validation, will you have demonstrated the sensitivity and specificity for the populations that the biomarker is intended for, right? Do you have the right clinical populations in your study design, and can you demonstrate that by the end of your proposal? And then will the biomarker be ready to use for the research or clinical community by the end of your proposal? Is this going to be a resource that others are ready to use? And with that, I think we can move on. This is the contact information for all 3 of us in the biomarker program, and a link to the website where there's additional information and some of the resources that Marianne discussed earlier. And we're happy to take questions.
Great, thanks, Carol and Marianne. This is Genevieve. Got some great questions coming in. First question, can a company of 3,000 employees apply for any of these grants with an academic partner? I'm going to go ahead and take that question myself. So a company with 3,000 employees would not qualify for the small business program options, STTR or SBIR. So they could potentially come in through the regular R61, R33 phased approach or the UL1. And there is also the possibility of subcontracting with an academic institution. So the next question I'll go ahead and throw this question to Marianne. Is there interest in a biomarker that provides the same or similar inference as a much more expensive to obtain biomarker, or is it more desirable to propose a biomarker that solves a completely unsolved problem?
So I think that depends on the state of that that particular disease and the therapeutics related to that disease. So if it turns out that the only biomarker that's available is something that really isn't feasible outside of research, then I would say absolutely it's worthwhile putting in an application for something that's more feasible and more easily used. If there's a biomarker already that exists, again, but only for use maybe in major hospitals, but you need something that's for point of care, again, that would be a reasonable application. So I guess the answer is it depends.
That's always the answer. Thanks, Marianne.
Yeah.
Carol, here's a question for you. For the R61, R33 preliminary validation in human subjects, is it allowed to enroll patients, or does it have to be pre-collected samples?
No, enrollment is certainly fine. That's perfectly within scope.
Perfect. Marianne, a question for you. Will the PIR 19315 consider a novel SNP as a biomarker for concussion-associated outcome?
So I'm not sure what FMP is, but definitely novel biomarkers are great, and we like to see those come in, especially if they fill a gap that exists in the disease area or the therapeutic area. So my guess is the answer is probably yes, but I got to admit I don't know what FMP is.
Great. Let's see. So Carol, if there's an existing biomarker but not a way to detect that biomarker, would a novel technique for detecting it fall into validation?
No, so the novel detection would need to go into the discovery stage or even the R01 space, right? The detection method needs to already be established for the validation stage.
Perfect. Mariana, on slide 16, when you were talking about the examples of activities that are out of scope for PIR 19315, can you elaborate a little bit on non-standardized sample sources?
Right. So one of the things that we really try to make sure people do is think about what their sample sources are, not only the sample sources, the sample collection, the sample preparation. We want to make sure that you can interpret your data, in other words. So if something is standardized, at least then the annotation for the for the patients themselves is all set up and done the same way. The way the samples are collected are all done the same way. That way, at least you've gotten rid of variables that could potentially really screw up the variability in your biomarker plan. So that's what we mean by standardized versus non-standardized. Often people use centralized, like a lot of disease communities have their own like clinical programs where they've got, let's say muscular sclerosis patients, they've got, TBI patients, whatever, and the hospitals have agreed to collect them in the same way so that the data can be interpreted. So, if you don't have access to something like that, what we try to suggest is that you do everything you can to standardize the collection method that you're going to use and show us exactly how you're going to do that. That has to be figured out and worked out before you can move to the analytical validation FOA.
Great. So Carol, the emphasis appears to be on single biomarkers. Would an application that attempted to validate an algorithm that combines several biomarkers, including a mixture of biochemical, imaging, and psychologic biomarkers, be responsive?
Yes, multimodal biomarkers are certainly within scope. It can get a little, hard to say biomarker or biomarker signature all the time, but yes, both of those are completely appropriate. It can add complexity to your approach, of course, and your validation, but they are certainly receptive to them.
Perfect. Marianne, what is the advantage of the discovery funding mechanism over the existing R01? The application seems to need quite a bit of customization for this funding opportunity, but the budget levels don't have a lot of difference.
Right. So I think in a way the advantage is more towards the general goal of the FOA and hopefully to the applicant who wants to actually deliver a candidate biomarker at the end. The FOA 19-315 requires you to do things in a very systematic and efficient way. So that you are pretty much assured you're going to have a biomarker that can go on for further validation at the end. So I think that's the biggest advantage in terms, you're right, in terms of the budget differences aren't really there. But it's, I guess you could say that in terms of funding and duration, they're fairly similar. But I think it's more the direction you get. The other thing is, because it's phase 2 and the milestones are part of this, you get a lot of interaction with the NIH team that will be working with you, who make sure that they have interaction with the FDA. So I think if you're not, especially if you're not used to developing biomarkers, this might be just an instructive way to do it, and maybe a more efficient way of getting what you want at the end.
Great. So Carol, is there interested in alternatives such as wearable devices that can deliver biochemical biomarkers that can quantify, for example, demyelination in MS or dopaminergic deficit in Parkinson's disease?
Yes, so that can be appropriate. You don't want a device development grant, but if it is really looking at a focus, then, as Marianne had said before, we're really agnostic to the modality of the biomarker or biomarker signature. So that certainly can come in, and it's good, this is general advice to everybody. If you're thinking about applying for the program, please do reach out to us, and we can have a call with ourselves and with, some of the scientific experts appropriate for your proposal, and sort of discuss more how it would fit.
Perfect. Marianne, do people need to wait to submit their grant application after they've received patent protection for the proteins that they've identified as biomarkers? Can you talk a little bit about the intellectual property?
So that's something that I guess is up to the strategy of the applicant. If you're concerned about, like letting any information go, then, and you want to have protection first, I guess that would be your strategy, then go ahead. But it's certainly most people who are applying for these biomarker grants, in terms of the intellectual property strategy, just show that at least they're able to operate, let's say, with the resources that they have. In other words, that, like, let's say that they're looking at a genomic biomarker, all the resources they need to develop that biomarker, they are free to develop. In other words, they're not infringing on anybody else's patent. So that's the type of thing. We usually ask for that type of thing in your summary related to intellectual property. If, you have a very clear plan to commercialize the biomarker, then providing us with a intellectual property plan in conjunction perhaps with your technology transfer office might be a good idea.
Perfect. Carol, can some of the funds from the grant be used to recruit more sample collection sites?
That, I'm not sure I know exactly what's being asked there. Marianne, do you?
So certainly, I guess maybe you're asking that if you come in on an application and you have a certain number of sites proposed in your application, but then you find that you may need more. That is, if that's what you're asking, then you can add more sites, but you usually can't change the budget at all. So you'd have to reallocate your budget to do that. But you know, in theory, as long as you were able to reallocate the budget, you could add other sites. And of course, you still have to, you know, deal with our single IRB requirements and, you know, make sure you have IRB set up for all these sites.
Thanks, Marianne.
Great, thanks. Either Carol or Marianne, can you give some examples of context of use?
Carol, do you want to take it or
Sure.
I can?
So, I mean, Marianne went over some of the examples, but you might have a diagnostic biomarker that differentiates subtypes of Parkinson's disease in order to help stratify patients for clinical trials, might be an example. Or you might have a pharmacodynamic biomarker to demonstrate target engagement of a therapeutic that you're also developing, as Marianne talked about a bit before. And, you know, basically, what's the biomarker category? What's the patient population it's intended for? What is the utility for clinical trials or therapeutic development, right? Those are the components. Marianne, do you want to give any more examples?
Yeah, and I guess the other, in addition to what Carol said, the other things that like apply to the actual use are, where are you going to use it? I mean, are you going to be using it in a clinical field setting? Are you going to be using it in a major hospital where there's a lot of resources available? That type of thing goes into it as well. So it's a lot of, I mean, you know, what are the resources available where you're going to use it? Again, as Carol said, what is the disease population? Is it a rare disease population? Is it an aged population? Is it a very fragile disease population? All these things that go into how practical is your biomarker to be used broadly by the community that you want it to be used for. So that's part of your decision, you know, how broadly will this be used? I mean, it could be used purely in, you know, like a single clinical trial. That would be all right too. But, you know, that's why it's important to outline very clearly what you intend right up front, because that does have a lot to do with how you're going to design your approach section.
Great. Carol, are consortiums eligible for these PIRs, and what if one of the consortium members is outside of the US?
So foreign components are eligible to come in. The application just needs to come from the US institution, but subcontracts or collaborations with foreign are certainly able to come in. So yeah, consortiums are welcome. And again, just a reminder about the $500,000 direct cost budget, because those consortium grants can be a little bit more expensive. You just want to make sure you reach out to us at least 6 weeks before so we can get approval. And you want to make sure that you can justify the necessity of the foreign component as well. That's just another part of the review process.
Great. Marianne, can you comment about cohort sizes that are reasonable for rare diseases? They're interested in CSF studies, but have difficulty finding more than 25 to 50 patients.
Right. So this is why I guess the quickest answer is it really, again, depends on what you need to interpret the study and what you need to provide confidence in the validation. So if it's a rare population that has a small number of studies, or it's difficult, you know, to get samples and all that, this is why it's very important to have a good statistician who understands biomarker development on your investigative team, because it'll be up to that statistician to provide a rationale for the ends and the power calculations that were provided.
Great. Carol, does the study need to be a multi-site study for the clinical and the analytical validation, or is one site sufficient?
So it's not absolutely required, but it really should be, unless there is an incredibly good justification of why it's not needed. And again, this just gets to making sure that it's demonstrating the reliability across sites and conditions, people, improve the reproducibility, all of those things. So yes, multi-site should be expected, but there are limited occasions where it might be okay to do a single site.
Perfect. Maybe this would be a good last question, Marianne. What's the best way for people to figure out which PAR their technology is at, and whether they should come in at the discovery or the validation stage?
So I think we've tried to make the entry criteria even more clear in the reissued versions that we're going to have for the validation FOAs. But bottom line is, if you have a biomarker, you've developed the detection method, and you have pilot proof of concept in terms of clinical validation, but you now need to do larger studies to analytically validate the detection method so that you're actually sure that your detection method has the right sensitivity and specificity for the entire diversity of the population you're looking at, then you're probably ready for the analytical validation FOAs. If you have not developed your detection method yet, then you should be applying to the discovery phase FOA. If you have, let's say you don't have really any pilot evidence that the biomarker has reasonable sensitivity and specificity from a clinical standpoint, then you should also be applying for the discovery FOA. If you want to apply for the clinical FOA, the clinical validation FOA, then we are assuming that you can show in your preliminary data a really solid evidence for the analytical validation of your detection method that will include all of the diversity of the population you want to look at. So you would have done something close to what we're asking for in the analytical validation FOA, and you would already have biomarker, you would already have pilot proof of concept, again, in humans, that the biomarker reflects what it's supposed to reflect. So hopefully that helps.
All right, I think we've come to the end of the webinar. For anybody's question that we did not get to, we have everyone's questions, and we will reach out to you individually to post the answers, or we will add it to our website when we upload this webinar. As I mentioned at the beginning, I will be sending out the recording and the presentation to everyone that has registered for the webinar. And just please reach out if you have any questions. But other than that, thank you very much for joining.
Thanks, everyone.
Thanks, everybody.